Welcome to Genomics Unlocked, the #1 webinar platform for all things genomics. Today's episode focuses on revolutionizing agriculture by harnessing the power of genomics, featuring Professor Robert Henry, Dr. Shangtong Li, and Professor Xiaolei Lu.
So, good day everyone. My name is Brett Verdeman, and it's a pleasure to be hosting you all today for our third webinar in the Genomics Unlocked series, where it's a series to basically offer a platform for knowledge sharing in all things genomics. So today I am very proud to invite Professor Robert Henry, Professor Xiaolei Liu, and Dr. Shangtong Li to speak to us more about harnessing genomics in agriculture, where we will explore the groundbreaking impact of NGS on the agricultural industry and discover how NGS technology is revolutionizing plant and animal breeding, and how genomics resources are a key platform on which agriculture improvement programs can be built. So before we start, we also have some upcoming webinars upcoming webinar in July, where we will also be talking about revolutionizing oncology with Alacra CMTA and MGI. So just thank you all for joining. For those of you that are not familiar with the GoToWebinar tool, so on your screen on your right hand side, you should see your GoToWebinar tool, where there will be the chat function and question function, for example. So if you have any questions, please just put your questions into the question box, and then it will be allocated to our specific speaker. So thank you all for joining today. And now I would like to introduce Professor Robert Henry, who is the Professor of Innovation in Agriculture at the University of Queensland, on the screen. So he will share a little bit more about what he does, but he conducts research on the development of new products from plants, and his research uses genome sequencing to capture novel genetic resources to improve the understanding of the molecular basis of quality of products produced from plants, and genome analysis to capture novel genetic resources for diversification of food and energy crops. So Professor Robert Henry, I'm proud to welcome you here to the Genomics Unlocked series, and I will give you access to your screen.
Thank you. So today I'm going to talk about building a genome platform for crop improvement using the example of the macadamia, but really also giving you many other examples that illustrate the power of this technology as a general platform for plant improvement. So it really is the case that genomics can revolutionize agriculture by harnessing the power of genomics across the entire diversity of the plants and animals that we use in agriculture. And in plants, we have a very large number of different species that we utilize that are important for food security and increasingly for energy security. And we need efficient systems to apply genomics to unlock their potential and to enable agriculture to cope with the future demands. One of the big areas that we need to be able to explore are the crop wild relatives. These represent a genetic resource, a reservoir of diversity that we should be aiming to capture using genomics and employ to deliver the food security that we need. In evaluating crop wild relatives, we can do this morphologically and look at some of the characters in them, but really we need to look at the gene level to be able to understand what they really offer, because quite often they don't appear to have the traits we want. We need to look behind their morphology and uncover the hidden information in their genes that we can use to deliver improved crops. I've worked across the whole range, really, of flowering plant diversity, looking systematically at sources of new plants that we might be able to employ in agriculture, and particularly in capturing much of the diversity around species like the ones listed here: rice, millet, sorghum, soybean, pigeon pea, mung bean, banana, grape, macadamia, eucalyptus, citrus, sugarcane. They all represent opportunities where genomics can deliver great opportunities for improving these as crops and improving the reliability of their production. And what we're really wanting to do is apply the advances in genetic technologies to this diverse array of crops to really not just be able to work with the major crops, but really to work with all of the minor crops as well, because they make important contributions, particularly regionally, to food security. And really, the big advance in technology that drives this is the advance in DNA sequencing. We all know that there's been a collapse in the price of DNA sequencing. And what is remarkable is that this is continuing even today, that we're seeing each year continuing drops in the cost of sequencing. And this is making sequencing accessible and available on a much larger scale and giving it much more power as a tool for agriculture. There have been advances in both short and long reach sequencing techniques, both in terms of the sequencing platforms, the assembly software, and what we now can do is achieve chromosome level of complex genomes from long read sequences alone. Not only can we sequence them, but today we now expect to get haplotype resolved genomes, gapless genomes, and these genomes are generally telomere to telomere. So for the first time, we have the complete genomes of species readily available by using this technology. We also have very effective short-reach sequencing platforms that allow us to do low-cost, high-quality resequencing. And this allows us to explore all of the diversity in our germplasm. The value of these better genomes and the pangenomes that we create by sequencing all of our germplasm is enormous. It provides important improvements in the understanding of the genetic basis of agricultural traits. It allows us to do selection in breeding by using fewer markers for genetic prediction and a much better chance of discovering the genes that control any trait of interest, because now we have very high quality genomes readily available for any organism that we choose. We've been involved in the last few years in progressively improving and evaluating different sequencing platforms and assembly tools for putting together the genomes of our major agricultural crops. And what we've seen is that as these technologies have improved, we've shown that by using sufficient sequence data, we can assemble longer and longer parts of the chromosome until, with adequate coverage, we completely sequence the entire chromosomes. And so what we now see quite typically from long-read sequence data alone is that we assemble a single contig that represents the chromosome. And these contigs have a telomere on each end, so they do represent the whole chromosome, including the centromere. So this is something that was really not possible only a couple of years ago. So it really does represent a major new opportunity for agriculture. And we're also now able to sequence all of our genetic resources so that we can build our agricultural genetic programs and our understanding of how to better manage our agricultural organisms on deep genomic data. We can resequence wild and domesticated germplasm using efficient whole genome resequencing. And what's important is that we can get high quality data at low cost, and that provides a really reliable resource that underpins all of our genetic improvement programs. I just wanted to describe a program where we're systematically looking at these horticultural crops. We're looking at many more, but these 10 are our priority, and give you specific details of the example of the macadamia, which is in the middle at the top there, as a species that really is benefiting enormously from this technology. macadamia is represented by 4 species, and this is a relatively recent domesticate, only in the last 100 years. The species are not particularly distinct, and so one of the great advantages of genomics is that we can actually identify the identity of much of the germplasm that is of unknown origin. We can correctly assign it to a species and much better manage the genetic resource that underpins the programs here. And we've used MGO sequencing platforms to really explore genomic diversity, phylogenetic relationships in both the wild and domesticated macadamia species. And this creates a powerful platform for macadamia breeding and macadamia production, understanding better how to manage the orchards. So these 4 species are the ones that we work with. We're interested in using the small nut species as sources of different plant architecture to produce more efficient orchards that produce the larger nuts. These species are all found on the east coast of Australia, but interestingly, they were domesticated by researchers in Hawaii and then reintroduced back into Australia in more recent times. We've now got high quality reference genomes for the 4 species, and this shows us things like an ancient genome duplication. 20 or 30% of the genome is actually duplicated, and this is really important insight for the breeders of a crop to have to understand the genetic structure of all of the species that underpin the domesticated crops and the relationships and the structures of their genomes. We've compared these and we've resequenced more than 300 genotypes that represent the germplasm resource that's available for these species. And in one case, we're now moving to sequence all of the known individuals of the species. So these species are rare and endangered, and in one case, critically endangered. So here is an example where we're using genomics to capture really all of the genetic variation that exists in the species. And ultimately, we will probably be able to say we have the sequences of all the alleles that exist in the populations. Using the MGO sequencing, we're able to resequence to aiming here. We're at 20x. You can see that we get quite consistent sequencing of at least 20x when we're targeting that here. So we've got very reliable data across the whole genome and can characterize differences in any of the genes in this germplasm. One of the ways we use this sort of data in plants is to analyze the chloroplast genomes. And the chloroplast genomes in plants can be assembled from short read sequence data to give a base perfect sequence, because all of our data sets where we sequence the total DNA from the plant, we're sequencing the nuclear genome to a target of 20x or more, but the chloroplast genome is sequenced to very high coverage, because for every copy of the nuclear genome, there's maybe 1,000 copies of the chloroplast genome. So we have data within the sample set that allows us to assemble and compare a base perfect sequence. This gives us a lot of information about the accession and is a really useful piece of information that complements our analysis of the nuclear genome. And here we show the nuclear phylogenies that we get when we look at the chloroplast and nuclear genomes. And what we see is that the chloroplast phylogeny is giving us a different evolutionary history to the nuclear phylogeny. And this is something that we see in many plant species that we see have different insights into the evolution. And to explain that a bit further, here we show the distribution in the wild of the chloroplast phylogeny. Here we have clades that show a geographic structure. So what we find in many plants is that the chloroplast tells us about the location in the wild from which the plant originated, because the chloroplast is very often geographically based. what this is due to is fairly simple. These are large seeds in these plants. The seeds aren't distributed very far, so the maternal genome stays locally and doesn't move. But if we look at the nuclear genome, we find a different result. So here we can look at the different structures within structures within the wild populations. Seeing the geographic structure in the chloroplast genome tells us the wild source or origin of a particular sample, and that's extremely valuable in collecting more resources and in utilising. We've done the same thing with citrus. We're just sequencing 11 citrus genomes, and this is providing a very powerful tool in the case of citrus to discovering the genes for resistance to the citrus greening disease, for example, that's causing major problems for citrus production globally. That we have found here in the wild many disease resistance genes that should provide resistance to this major disease and really ensure reliability of citrus production globally. We have similar potential in bananas, where the wild species have resistance to the new fungal diseases that are threatening production in many locations. We're similarly applying this approach to mango, where we've resequenced very large germplasm collections. And what we've discovered here is that many of the wild species seem to be hybrids with domesticated mango. So here we're learning that over the long period of domestication, there's been gene flow in both directions, from the wild to the domesticated gene pool, but also back the other way, so that many of the wild species, as we now know them, have an origin with maybe one of their parents being a domesticated mango. So this is something that we also see in more and more species when we come to be able to sequence all of the genetic resources at the whole genome level. So we're currently sequencing large germplasm collections in all of these species. We've sequenced all of the avocado varieties. We're sequencing bananas. We're sequencing all of the diversity in these important crops. We're also beginning to look at the pulses. There's real opportunity for applying this to the great task of producing more protein from plants, which is really an important new area. We've discovered, for example, wild species that have resistance to the major insect diseases that limit the production of some of the grain legumes, and so genomics is really going to enable more stable production of these important pulses, and to characterise some of the wild species. Here's one that's growing in the desert that has great resistance to drought, and again, sequencing these genomes provides great new insights into how to improve crops for the future. We've even applied sequencing on a large scale to coffee, and really this is helping us to understand coffee quality and how we can meet the challenge of breeding coffees that will survive in the climates of the future, where we're seeing climates that are less favourable for producing high quality coffee, but where there will be ongoing demand for the product, and we require new varieties by sequencing very large numbers of genotypes. We've found some of the genetic variation we need to address the important future of coffee as a crop. This includes working with many of the wild relatives of coffee that represent new genetic resources. We're currently looking at even wider applications, where we're looking at understanding the origins of yams and really supporting some of the insights there. And in many species, and blueberry is the example I would cite, we're using now this for intellectual property protection. Breeders can protect their varieties, can understand the source of germplasm that's being used by their competitors by using sequencing to really provide that intellectual property protection. Of course, knowing the genome sequence precisely allows us to edit it. And that's the other great application of large-scale genome sequencing to characterize germplasm to the extent that we can make highly targeted edits and that we can characterize the products of that editing and ensure that it's safe and meets regulatory requirements for release back into the environment. And we're seeing the potential for large-scale application of sequencing to the deregulation of gene-edited plants for agricultural and food applications. And beyond applications of genomics to support food varieties, we also see applications in related areas in producing energy crops, which is increasingly an important issue, but also in supporting the conservation of biodiversity by analysing wild populations that are being conserved for environmental reasons. So genomics with these technologies, exactly the same approaches really, can support plant breeding, biodiversity conservation, and can support both food and industrial uses, of course, of our crops. So we're busily looking at capturing biodiversity for food and energy crops, both by developing new crops, but also by supporting the development of new crops by undertaking what we call de novo domestication, where we take a species that was never domesticated. And by using sophisticated genomic analysis, we can develop a plan to domesticate a completely new crop that provides a new opportunity for agricultural food production. And this is a very exciting area that's driven by our ability to sequence very large numbers of samples. So I'll just finish by thanking the many collaborators in the work that I've been talking about, and thank the many people in the lab and the researchers that have been collaborating here. And I hope I've shown some of the some good examples of how large-scale genome sequencing can really revolutionise agriculture and really provide the key platform for crop improvement. Thank you.
Great. Thank you very much, Robert, for that enlightening talk and providing us with more information on how genomics can be a key tool to support selection of germplasm in breeding through the case of macadamia, and how we can adopt similar approaches for many other crop species. So thank you very much for that. I would now also like to introduce Professor Xiaolei Liu, who is the professor at Huazhong Agricultural University, specializing in statistical genetics and genomic breeding. So his research focuses on statistical genetics, algorithms, multi-omics integration, knowledge bases, and intelligent phenomics studies. So we're excited to learn more about his developed algorithms that have been published in multiple articles. So welcome, Professor Xiaolei Liu. You may share your screen.
Okay, thank you. Good morning, good afternoon, respected colleagues and friends. My name is Xiaolei Liu from Huazhong Agricultural University. Today, I'm very glad to have the opportunity to present our recent work. My presentation title is High BLAP Integration of Statistical Methods on the BLAP Framework for Efficient Genetic Evaluation using Big Genomic Data. The breeding plays a remarkable role in feeding the world. The breeders select the superior individuals in each generation of animals and plants, driving the gene frequencies of populations towards the directions for human needs. For instance, in the wild boars, we can see that the head comprises 70% of the body, while the meat-producing body parts only accounts for about 30%. However, through generations of selection, we can see that the modern domestic pigs have completely reversed these proportions, with the meat-producing parts now is exceeding 70%, and the head only accounts for 30%. This transformation also observed in plants such as significant increases in yield from testing to the modern corn. In simple terms, that breeding is a process of selecting the best individuals among many to choose the cream of the crop. The breeding history has been closely associated with the development of individual ranking techniques. Like in the early days of phenotypic selection, the breeders ranked individuals based directly on the phenotype values, the trait values, keeping the superior ones and eliminating the inferior ones. However, the breeders later realized that although some individuals showed excellent performance in terms of the production traits, their parents, siblings, or progenitors didn't exhibit the same level of performance. So the wise breeders pointed out that the phenotypes are influenced by both genetics and environmental factors, and can be further divided into the breeding values and residues. So if the breeding values can be accurately separated from the phenotypes from the residues, the ranking of individuals will be more precise. So therefore, the plant breeders utilize the same with the consistent DNA and conducted multi-year, multi-location trials to partition the variances of breeding values and environmental effects using the mixed linear model. Meanwhile, the animal breeders incorporated the pedigree information in addition to phenotype information and used mixed linear model to separate the breeding values and residues as two random effects, thereby partitioning their variances from the phenotypes. Thus, the breeding entered the era of EBV, which represents the estimated breeding values. Since the commonly used methods to solve mixed linear model for calculating the EBV is blob, then we always call this era is the blob era. With the advancement of the sequencing technologies, we see that a small number of molecular markers associated with the breeding objective traits were added to the blob model, further enhancing the breeding efficiency. But we see that the marker effects on the complex traits, like the production traits, are always not that significant. And the markers with high effect tend to quickly become homozygous in the populations. The addition of 3 to 5 markers doesn't help to improve the breeding efficiency a lot. At this point, the genomic selection emerged. It aims to accelerate the genetic improvement by improving the EBV accuracy and shortening the generation intervals to speed up the genetic progress. We can see that as the breeding progresses, more and more information is being utilized, from using only the phenotype data to phenotype data combined with the pedigree data, and then to phenotype data combined with pedigree and genotype data. The statistical models are needed to effectively utilize this increasing amount of information. And this process is accompanied by the development of the statistical models, particularly with the widely applied blob framework models in the breeding history. The blob model utilized 2 essential pieces of information. the phenotype information and the genetic relationship among the individuals. Starting from the P-blob model, the pedigree information is used to construct the relationship matrix, which we always call it A matrix. With the addition of the genomic information, Montoni simple error are reduced, leading to more accurate estimation of the genetic relationships between individuals based on the genomic data, which we call it the G matrix. This improvement also enhances the EPV accuracy. In cases where the genomic information is not available for all individuals in the pedigree, a mixed relationship matrix, we call it H matrix, can be constructed using both the pedigree and the genotype information. We use the we call this single-step G-blob. We use this model to maximize the utilization of the breeding data in a breeding farm. The use of a larger and more comprehensive data sets can result in higher accuracy and give us the accurate EBV. However, whether it is the A matrix, G matrix, or H matrix, all of them display the genetic relationship between the individuals. Regardless the breeding objective trait, the same relationship matrix is used for all different kinds of types of traits. Several studies also have shown that constructing the trait-specific relationship matrix can further enhancing the EBV accuracy. And these methods aim to utilize not only phenotype, pedigree, and genotype information, but also the additional information about the associations between genotypes and phenotypes. So under the block framework, the optimization of model the algorithms has always focused on the efficiency and accuracy. In terms of efficiency, there are two aspects. First, it involves reducing the computational complexity of the algorithm models to improve their ability to handle the large data sets. Secondly, it aims to maximize the utilization of the limited computational resources, such as the memory and CPU. In terms of accuracy, there are two main considerations. Firstly, it involves enhancing the algorithm ability to utilize the information effectively. For example, it explores whether this algorithm can effectively incorporate the multi-omics data or high-dimensional phenotype data to improve the EBV accuracy. Secondly, it aims to strike a balance between the efficiency and the accuracy. Next, let's take a look at the mixed linear model and the blob. So the mixed linear model utilizes the phenotype data, environmental factors, and the relationship matrix among individuals to calculate the EBVs. The blob method for solving this model was originally proposed by Professor Henderson from Cornell University around 1950. But the widespread application of blob calculation began in 1970s. The blob solution for the entire equation can be written as this formula, where the left-hand side matrix is inverted and multiplied with the right-hand side matrix to solve for the fixed effect, the B, and the random effects, the U, in the middle. The most complex calculation involves in this process includes the genetic parameter estimation to calculate the lambda in the middle red box and also compute the EBV. For the lambda, the genetic parameters the algorithms, we always use the Ramal method to calculate the lambda. So it involved inverting and computing the covariance matrix, requiring the multi iterations and high computational complexity. As a result, the update frequency for the genetic parameters is often low, typically maybe every half a year or once a year to update the lambda information. So in the EBV calculation part, it involves inverting the relationship matrix K, also in the middle red box, which could be a matrix of maybe 10,000 individuals. It required to invert a 10,000 by 10,000 matrix. So additionally, inverting the entire left-hand side matrix, which includes the K inverse, is even larger. EBV calculation is a task performed by the protein programs on a regular basis. In the context of the limited computer resources at that time, thanks to Henderson's very ingenious algorithm for inverting the pedigree-based relationship matrix and the use of sparse matrix calculation strategies, the efficient solution of the mixed model equation was achieved. The breeding computation research team led by Ignacy at the University of Georgia developed the FSPK algorithm to efficiently solve the large sparse matrix inversion in the mixed model equation. So the widely used the software tools in animal breeding, such as the DMU in Europe and BLAPEP 90, both of them rely on the FSPK for efficient calculation for the EBVs. But with the integration of the very big genomic data, the relationship matrix between individuals has changed from the A matrix to the H matrix. And the inverse matrix of the relationship matrix has changed from very sparse to increasing dense. In the diagram, we can see that the black blocks in the H inverse matrix represent the dense regions. As the number of genotype individuals increases, the black blocks gradually spread to the from the upper, from the left to the corner, from the corner. The FSPK-based sparse matrix strategy struggles to handle this increasingly dense and is no longer suitable for the genomic breeding computations. So we see that the FSPK algorithm faces the challenge of increasing dense in the non-zero elements of the relationship matrix H as the number of genotype individuals grows. And this leads to a dense left-hand side matrix. The question is that how can we efficiently compute the inverse of the left-hand side matrix? And to address this issue, the same research team, Ignacy team, proposed the Llamas algorithm. The Llamas algorithm follows the similar strategy of solving the mixed model equation. It automatically identifies the dense blocks within the matrix and performs the blockwise decomposition and inversion operations. By combining the Llamas with the MKL method library, the efficiency of the computing the inverse matrix of the left-hand side matrix is significantly improved. Let's see the second challenge. The dimension of the G matrix is also keep on increasing as the number of genotyped individuals grows. The construction of the left-hand side matrix requires the inverse of the relationship matrix, which can be either the H inverse or G inverse. Efficiently solve this inverse matrix is the other challenge. Also, the same team a solution was proposed by dividing the genotyped population into two groups. They called the core group and the non-core group. Only the inverse of the relationship matrix for the core group needs to be computed, avoiding the need to calculate the inverse of the entire population. So this approach further reduces the computational complexity for calculating the J inverse. However, we see that the proportion of the genotyped individuals is rapidly increasing. Taking the big pig breeding companies such as PSA and Danbreed as examples, the number of genotyped pigs per year has reached hundreds of thousands. As a result, the matrix becomes even denser. To address these issues, we explored the use of algorithm based on the variance covariance matrix. And this approach is based on the variance variance decomposition of the phenotypes and estimates the various components and breeding values. Therefore, it is also known as the V matrix strategy. In the process of estimating the variance components, only the inverse of the V matrix needs to be solved without needs to compute the inverse of the whole relationship matrix. So in the process of the EBV calculation, if the various components are unknown, the inverse of the V matrix needs to be solved. However, if the various components are known, we can use the iterative strategies such as the PCG to compute the PY based on VY, avoiding the direct inversion of any large matrices. So compared to the strategy based on the mixed model equation, we call it MME. The V matrix strategy has the lower computational complexity. It is dependent on the number of individuals that has phenotypic records and doesn't require the inversion of any whole relationship matrix for all the individuals. On the other hand, the mixed model equation strategy is dependent on the product of the total number of individuals and the number of random effects, requiring the inversion of the relationship matrix for all the individuals. So as the number of random effects increases in the breeding value evaluation model, the left-hand side matrix grows exponentially. Additionally, both the V matrix and the left-hand side matrix have a probability being non-invertible. However, in most cases, we see that the V matrix is always positive definite, making it more likely to be invertible compared to the left-hand side matrix. Therefore, I believe that the computational strategy based on the V matrix is more suitable for denser matrix calculations in the era of genomic breeding. However, here we still need to calculate the inversion of V matrix and how to find a solution to avoid all the large matrix inversion is very important if we have a ultra large scale the genomic breeding data in the various components estimation and EBV calculation. We came up with the HE regression algorithm, which was first proposed by Hisman and Elstone in 1972. This method establishes the regression equations between the similarities in phenotype values and the corresponding elements in the pedigree-based relationship matrix. The regression coefficients represents the estimated probability, and the computation is simple, efficient, and does not involve any large matrix inversions. we apply this method to the various components estimation in the genomic breeding, and expand this method to accommodate for multiple random effects models for the genetically correlated trees. Using the genetic parameters calculated through the HE regression, we developed a PCG solver specifically designed for the dense matrix. By combining the Intel's MKL math library and the Huawei's Kunpeng KML math library, we can efficiently perform the EBV calculation, completely avoid the large matrix information, the large matrix inversion. So our new strategy is HE regression plus PCG to avoid all the large matrix inversion in the whole genomic breeding calculation process. To make a short summary from the era of the traditional breeding to the genomic breeding, there has been a transition and a development in the matrices used for genetic evaluation. These matrices have evolved from being very sparse to gradually becoming dense. With the rapid development of the genotyping techniques, such as the MGI sequencing, the cost associated with the genotyping is decreasing rapidly, leading to completely dense matrix in the future. Correspondingly, the computational strategies have shifted from solving the equations based on AI-RAMO plus mixed model equation to using the AI-RAMO plus V-matrix strategy, and finally to the HE plus PCG strategy, which avoids any large-scale dense matrix inversions. In simple terms, the classic software tools like BLAP590 and DMU are suitable when a small proportion of individuals in the population have been genotypes, for example, less than 10%. On the other hand, HiBLAP is more suitable when a large proportion of individuals in the population have been genotyped. When the proportion of the genotyped individuals increases, the computational advantages of HiBLAP compared with the existing tools become more pronounced. The HE algorithm can also be combined with the classic AI-RAMO algorithm to balance the efficiency and the accuracy of the genetic parameter estimation. So therefore, we choose the first letter from HE, the second letter from AI, and added BLAP to create the name HiBLAP for our tool, which is called Tianquan in Chinese. HiBLAP is based on the new strategy that's different from the FSPK, and it covers its function covers the entire process of the genomic breeding. It is suitable for the single treat, multi-treat, and repeated record treats. And the model allows for the flexible addition of the covariates, fixed effects, and the random effects. And it can construct the reshape matrix based on the pedigree or genotype or both information, and utilize multiple variance components algorithm and their combinations to estimate the various components. HiBLAP can be used for genomic selection and genomic mating calculations, including the EBV calculation and the marker effect estimation. And the software employs the techniques such as mixed precision computation and memory mapping techniques to reduce the memory consumption during the entire calculation processes. Additionally, we have implemented accelerating using multiple math libraries for different computing processors. We have compared HiBLAP with the internationally renowned the existing breeding software tools such as BLAP90, ASRAMO, and also the human genetics analysis tools, JCTA. HiBLAP shows significantly computational speed advantages in aspects such as constructing the reshape matrix among individuals and the various components estimation. Moreover, we see that as the number of random effects increases, the computational speed and the memory advantages of HiBLAP would become even more pronounced. As the proportion of the genotype individuals in the population increases, the computational advantage of HiBLAP becomes bigger. We can observe that when the proportion of genotyped individuals is low, such as less than 10% or around 10%. BLAP90 with the FSPK algorithm strategy is very suitable for computing this type of data. It's get the very fast computation speed and utilize the smaller memory. However, as the number of genotype individuals increases, the computation time for calculating the EBV in BLAP90 significantly increases. In contrast, for the same data size, the computing time of HiBLAP remains nearly unchanged. Additionally, we evaluated the HE plus PCJ strategy using the data at the scale of the UK Biobank, which consists of 500,000 individuals with 1 million markers for each. We were able to complete the estimation of the genetic parameters and additive genetic values within 1 hour using this strategy. Furthermore, the results were accurate without any loss of information. Our final goal is to apply the developed algorithm and the software tools to the practical breeding. However, the breeding operations not only require HiBLAP, it also requires an integrated solution that covers the data collection, individual selection, and mating guidance. In addition to this breeding calculations provided by HiBLAP, this integration solution should also include the functions of data collection and management of the pedigree, phenotype, and genotype data. To facilitate the breeding calculations, we have designed the concept of populations, which consists of the same breeding lines from multiple specified subfields in a breeding company. The populations allow for efficient organization of the genotype, phenotype, and pedigree data, enabling efficient data access during the breeding calculations. We have developed automated and efficient methods to handle the gene chip data with varying densities and formats. Additionally, we utilize the genotype data to automatically correct the errors in the pedigree and implement the quality control measures on the phenotype data based on some predefined standards. We perform the genetic parameter estimation, breeding value evaluation, and the composite selection index calculation at the population level. All of these calculations are driven by a configure file, which specifies the required data files, locations, and the file names, formulas for calculating the composite selection index, and also the EBV models, the relevant EBV models. Here is an example of a configure files which includes all the necessary information for data extraction and the model settings. It allows for easy specification of the covariates, fixed effects, random effects, and the genetic parameters. In order to integrate high blood and the various breeding data management modes into existing production operation management system as the breeding companies, we have packaged them as the API format. By integrating the API, the genomic breeding calculations can seamlessly connect with the overall information flow of the production operation management system. It enables the third-party information platforms to leverage the genomic selection and the genomic mating functions by exchanging the data through parameter-based communication. And for the breeding farms that require integrated solution for the information management and the genomic breeding, we provide a comprehensive solution, and it is suitable for the big pig farms management, genomic selection, genomic mating for efficient commercial pig production. And this work was completed by the pig genomics and breeding team led by Professor Shu Hongzhao in Huazhong Agricultural University. The main developers include Dr. Yin Lilin and PhD candidate Zhang Haohao. The project also received some funding from the National Key Program for Young Scientists. And also you can download and use HiBLAP for free by visiting the website or scanning this QR code. We greatly appreciate the valuable suggestions provided by the HiBLAP users. Okay, that concludes my presentation. Thank you.
So thank you, Professor Xiaolei Liu, for presenting us with these tools that have been effectively used in agricultural genetics and breeding research. So I would now like to introduce Dr. Shangtong Li, the CEO of Glibizzia Biosciences, who aims to develop affordable genotyping technologies and efficient phenotype prediction tools to make life predictable. So I'm going to share.
Dear all, thanks for the invitation by MGI and the kind introduction of the host. This is Shangtong Li from Glibizzia Biosciences. I am very glad to share some of my understanding and efforts in accelerating breeding by multi-scale sequencing technologies and the accompanied computational pipeline. Title for my presentation is Multi-Scale Sequencing Technologies to Make Life Predictable. We humans think we are the host of the Earth. However, our beautiful life needs the company of thousands of species, especially the agriculturally important species and the pets. Our ultimate goal on accelerating breeding is to make life of various organisms predictable from genotype data, including crops, livestock, and pets. Based on the MGI sequencing platform, we are developing versatile sequencing tools and computational pipelines to achieve this goal. In sequencing tools, we tried to provide affordable and scalable technologies, including whole genome sequencing at different coverages, targeted region sequencing, and multi-omic sequencing, including transcriptomic and epigenome sequencing, to enhance the mechanistic understanding of life. We also collaborate with many researchers in China to accumulate organism-wide dynamic single-cell sequencing data. And on the data analysis part, we develop pipelines from the very basic genome assemble, population genetics, and to apply the data for a breeding platform that could perform genome-wide association study, genome selection and design breeding by integrating big data. Humans have a long history in breeding to feed ourselves. Based on the techniques involved, the historical progression of breeding could be split into 4 major stages, with the breeding efficiency increase in each stage due to the advances of available tools. The first stage of breeding began 10,000 years ago, as people around the world domesticated and selected wild plants and animals for food by experience. In these stages, professional breeders did not exist, but phenotype-based selection by local, independent farmers eventually resulted in the dramatic phenotypic changes of these plants and animals. But taking the timescale into consideration, the first stage of breeding is low efficient despite a great improvement in feeding ourselves. And the second stage of breeding began in the early 20th centuries, when some basic biological principles were recognized, such as the inbreeding depression, Mendelian genetics, and the quantitative genetics theory. In these stages, we could create new materials by mutagenesis and hybrid, and select the plants and animals by field trials, controlled crossings, and statistical analysis, and provide precise measurement of yield at scale. While in the past 2 decades, we entered into breeding, the third stage of breeding, when molecular markers and the genomic data begin to complement phenotypic data. This stage started with marker-assisted bycrossing and pedigree confirmation, then moved to dissect the complex traits with linkage mapping. The introduction of high-throughput genotyping then expanded the quantitative genetic toolkit to dissect variation in nature populations and to evaluate breeding values by performing genome selection. And we are now on the cusp of the 4th stage of breeding. Although in my opinion, we have not stepped into the stage for any organisms. But all of us will believe such a scenario will be ideal for breeding. In this scenario, functional genetic variants can be rationally combined both faster and better than ever before. And genome resequencing studies can now cost less than genome resequencing studies can now cost less than a replicated yield trial, and genome editing is expected to enable parallel process modifications of hundreds sites per generation. And machine learning approaches enable the processing and the interpretation of agronomic data at a high accuracy. We will be equipped with the powerful AI system to integrate the big data and the knowledge to create an ideal breed. And now we are on a time when various molecular breeding techniques are developed at different throughput and cost. With the increment of genome coverage, the cost per sample increased, and the PCR-based genotyping method could detect variations in several to 100 loci, and the targeted sequencing could detect variants for 100 to 50,000 to 15,000 loci. While whole genome sequencing could detect variants not limited by number of locus. And in the following slides, I will go into some of the details later. There are 3 keywords for genotyping technologies in agricultural applications. The first is the large sample size. Usually more than 1,000 samples were kept for each generation in a breeding population. And the second keyword is cost. The budget for each individual is lower in genotyping than phenotype measurement, so that the breeder will have a strong wish to use sequencing data to predict the phenotype, well, instead of a direct measurement of the phenotype. And the second keyword is timing. The quicker of a breeder could predict the future phenotype, the more field, space, and nutrition will be saved for keeping a large number of plants or animals. To feed the above keywords, we have summarized several essential points for genotyping technologies in agriculture applications. Firstly, the sequencing results must be accurate to facilitate downstream phenotype prediction. And the price of test must be affordable to make it possible to include a large number of samples for precise phenotype prediction and subsequent selection. The prediction of phenotype must be achieved in a short time, best at 1 week, to save time for progeny selection, and the loci in a sequencing panel should be scalable to add and remove variants so that the genetic basis for prediction could be modified with the accumulation of the data. So in summary, a good sequencing technology should genotype as more loci with phenotype predictive power as cheap as possible, as accurate as possible, and as rapid as possible. Under these points, I would like to introduce two genotyping technologies that have been adopted in genome selection and I think will be more and more used in the coming years. The first is target sequencing technologies, either by an applicant-based assay or by a hybridization capture-based assay, usually called a liquid chip. There are several advantages for the liquid target sequencing reactions. Firstly, although there are many genomic variants in a breeding population, most of the genomic regions are invariant in a given species. Target sequencing could obtain the genomic variants on a breeding population without missing many genomic variants. And as the target sequencing only covers a small fraction of the genome, we could sequencing these regions to a very, very high depth to enable accurate variant calling without a very high sequencing cost. So the target sequencing technology are affordable for most breeders compared to the whole genome sequencing technologies. At last, compared to solid SNP array, the target sequencing panels is very flexible in the production. We do not need to order a large amount of reactions and are relatively easy to add a novel interesting region into the panel. Well, a second promising genotyping technology is the low coverage whole genome sequencing. And in the low coverage whole genome sequencing, after building up a reference panel by high coverage whole genome sequencing data, we could sequence each individual in the same population with a very low sequencing coverage on the whole genome and calling the variants accurately by genotype imputation, so that the cost is much more affordable compared to the high coverage whole genome sequencing. To enable genotyping in a large sample size, a significant advantage of the low coverage whole genome sequencing in breeding is that they cover the whole genome to identify novel variants associated with any interesting traits, which is impossible for targeted sequencing. In addition to the two genome sequencing technologies to meet the requirement of the fourth stage of breeding, mechanistic dissection of molecular networks with multi-omic data and the interaction of different genes, RNAs, and proteins. These large integrative data are required to rationale design of the genetic components of a plant or animal. Here we show a subnetwork for maize flower timing-related genes, a maize network map that contains genomic, transcriptomic, and proteomic network across across maize development have more than 1,000 such subnetworks, and these networks will be tremendous resource for rational design of an ideal maize. As an organism is constituted by many different organs and cell types, high resolution profiling by single-cell level sequencing at high spatial resolution also provided unprecedented view and opportunities for precision breeding today. And next, I will discuss two cases in designing breeding program to accelerate the progress to obtain a desired phenotype. One case is to measure the qualitative trace loci with a convenient and cost-effective multiplex arms PCRCE panel. And the other is the case to show rational selection of the parental pairs that will benefit for acceleration of the breeding progress of rice yield traits. Here I show 7 kites that have very similar face but differ greatly in their coat color. An interesting story is that these kites with different coat color have dramatically different price. That means very different margin for filling breeder. The good news is that these colors are controlled by about 10 loci, including the brown locus, the dilution locus, and so on. Without a genetic test, a breeder may take about 5 to 10 years to obtain a desired color. And with genetic test by selecting proper kites with recessive locus, a breeder could obtain a desired coat color in 2 to 3 years. In addition to the color loci, the filling breeders also care about the blood type and the common genetic disease locus, which may cause severe health problems to their kites. So the breeding goal of the filling breeder is to combine about 10 beneficial alleles and at the same time avoid more than 10 low-frequent deleterious loci. To meet the requirement of the genetic test on about 20 loci at an affordable way, we resort to a smart PCR-based genotyping method called ARMS, which is sought for the application refraction mutation system. Here is the diagrammatic presentation of the tetraprimer ARMS PCR method for SNP identification. Briefly, 2 allele-specific applicants are generated using 2 pairs of primers, one pair producing an applicant representing the G allele, and the other pair producing an applicant representing the A allele. By positioning the 2 outer primers at different distances from the polymorphic nucleotides, the 2 allele-specific applicants differ in length, allowing them to be discriminated by gel electrophoresis. And we further apply the ARMS strategy to the capillary electrophoresis system, which have a very high resolution in discriminate different applicant size and is capable with fluorescent detection, so that we could detect multiple SNP size in a single tube using the combination of different primer pairs with similar amplification efficiency. And this convenient and cost-effective genetic test could cover 1 to 10 and maybe 20 or 30 sites at the same time. And this test has been sold to thousands of filling breeders all around China, and hundreds of tests were performed per day now in our lab. And the multiplex arms PCRCE approaches could also be applied to any circumstances that require genetic tests on a series of qualitative loci. Now we have expanded this approach to various applications, such as the livestock genetic lineage correction by amplifying specific STR locus, and the plant seed purity test during seed trade to avoid any fake seed, and screening potential genetic defects in cattle with our collaborators in several different fields. And a second case I would like to share is a proof of concept of a parental combination selection strategy on rice to improve the per plant yield. The rice yield is a quantitative trait, so that it is a more complex case. We integrate pedigree simulation and genome selection on target sites to select the best parental combinations and to test whether this is better than the traditional genome selection approaches. So what is parental combination test? In a common genome selection procedure, given a reference population, here take the rice as example. The goal of the genome selection is to select parental lines with highest GEBVs. However, some of the parental lines with high GEBV value may not be complementary to each other in combining beneficial alleles. We have mentioned that in the fourth stage of breeding, a major goal is to combining many different beneficial alleles into one breed. So we ask if we change the goal to individual selection to parental combination selection, which select combinations that produce higher median or supreme GBVs in the simulated progenies. If this kind of selection will accelerate the progress of obtaining an ideal breed. There are several challenges in selecting proper parental combinations on accelerating breeding on a quantitative trait. Firstly, the simulation of hybrid and prediction of genotypes in progenies. Here we solve the problem by a powerful pedigree simulation software called Pedigree Sim, which could not only simulate the genotype of progenies of diploid, but also the tetraploid. And the second challenge is the large sample size for computation. If we perform a genome selection on a breeding population with 1,000 individuals, we need about 1,000 phenotype predictions. However, if we would like to test the parental pairs in the same population, the times of prediction increases to about 300,000. Here we reduce the computational space by targeted selection on unlimited number of sites and by using a computational efficient framework called LET-GBM. And the third challenge is to combine the beneficial allele and avoid the deleterious mutation at the same time in one breed. A good news on the rice yield case is that rice is not only an agriculturally important species, but also a model plant with sufficient basic researches to identify numerous functional genes. We manually select about 300 QTLs covering various functional traits on rice, including about 80 QTLs directly and significantly associated with rice yield. And targeted selection on this loci will greatly reduce the computational space on prediction the yield from genotype data. We also adopt an efficient computational framework called LET-GBM. It has several advantages compared to other GIS algorithms. It has a faster training speed with lower memory usage with a better accuracy. It supports parallel, distributed, and GPU learning, and it is capable of handling large-scale data. And LightGBM has been shown to be powerful in genome selection in crops in a publication on genome biology in 2021. Firstly, we examine the importance of our selected QTLs on rest yield. Surprisingly, we found a number of beneficial alleles in each rest line positively correlates with the rest yield. Here, each dot represents one rest line. And this figure means that the 300 loci we have selected are important for rice yield, even without the machine learning-based prediction. Next, we tested the yield prediction power of the 300 functional QTLs under the LightGBM computational framework. The Pearson correlation of predicted yield approaches to the real yield in the training set after about 10 iterations. It also shows a very great predictive power for the test set, suggesting that we could accurately predict the yield of a rice progeny to evaluate the combination of different parental pairs. Noticeably, the predicted power may decrease if the training set and the test set are under very diverse genetic background. With the accurate prediction of rice yield by limited number of QTL sites, we have simulated 2 potential applications to show the power of parental combination selection. Firstly, given a reference population, we compared the parental combination approaches by selecting complementary lines in beneficial alleles, and to random selection of parental lines by GEBV, and predicted the yield improvement following hybrid and backcrossing for several generations. Here, each line represents the yield improvement roadmap following backcrossing with parental line. And we found that parental combination selection approach saw about 20% increment of predicted yield compared to the random selection approaches. In another scenario, if we have a candidate rice line for improvement and a candidate donor parent population, we tested the selection of complementary parental combination selection of the donor lines with the GEBV selection of the donor line and with the supreme phenotype selection of the donor lines. Again, the complementary parental combination selection approaches represented by the green line have a much better maximum yield after several generations of backcrossing. As we have shown the power of using limited number of sites in predicting a quantitative loci, now we are collaborating with MGI on an applicant-based target sequencing technology called Etoplex to develop various panels. The Etoplex have several advantages, including primer auto-design, low amount of sample requirement, operational convenient, and fast in library construction. We hope we could have more and more target sequencing panels like the rice yield case that could predict life with an appropriate number of genomic variants. To summarize in my talk, I will talk about versatile sequencing technologies that were available now to accelerate the progression of molecular breeding. And the application of breeding, the third stage of breeding and the efforts to the fourth stage of breeding requires appropriate technology selection, taking multiple essentials, including cost, speed, and flexibility into consideration. And in the quantitative trait case, we have shown that parental combination selection with computational resource saving approaches may accelerate the progress to obtain an ideal breed. And thanks for your attention, and any questions are welcome.
Great. So thank you to Dr. Shangtong Li for showing how NGS-based technologies offer new opportunities for plant and animal breeding. So yeah, thank you to all our speakers. The floor is now open. So we have received a few questions. Professor Xiaolei Liu, has this high bulb algorithm been applied in practice, and is it okay in the practice? If you don't mind answering.
Okay. Yes, the HiBLUP is actually a software tool. It has been applied in several big pig breeding companies in China so far. Yeah.
Great.
It can be applied in practice.
And what about the hardware requirements for data analysis?
Okay, HiBLUP works on most hardware platforms that I have known, and the detailed requirements for the hardware depends on the data scale, such as the sample size, number of individuals with phenotypic records, and the number of genotype individuals. Yeah, if you want to use HiBLUP, you give me those information, then we can calculate how big is the memory requirement, and how many CPUs we need to calculate that data. Okay, thank you.
Right. So I think there's another question for you. What is the difference among low coverage whole genome sequencing array and liquid chip data when using HiBLUP analysis?
Okay, thank you for the question. Actually, that we use HiBLUP to do the genetic evaluation, because HiBLUP uses the phenotype information and the genetic relationship matrix. So there's no difference for you to use different types of genotype data. The only difference is how do you organize the genotype data format, how to prepare the genotype data when you use the the whole genome sequence data or some array data.
Okay, well, there's been quite a few other questions, but I see they've been answered. So thank you, everyone, for joining today. And I'd also like to highlight that we do have an upcoming webinar on the 5th of July. And we hope to see you at the upcoming webinar. So thank you very much for joining. Keep well, and goodbye for now.